Namaskar and good morning to all of you. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. In this series of lectures on archegoniates, we have till now covered a long way. We have seen various biological aspects of the group bryophytes, followed by pteridophytes, and now we have already entered into various introductory aspects of gymnosperms. As far as gymnosperms are concerned, we have gone through the life cycle of gymnosperms in a very brief manner. Today in this lecture, I would be covering the reproductive aspects of gymnosperms. So as far as reproduction in gymnosperms is concerned, it takes place through asexual mode as well as sexual mode. Although the asexual mode of reproduction in gymnosperms is very rare, but nonetheless it is shown by some of the species such as cycas. Vegetative methods of reproduction are very uncommon, but in case of cycas, they do produce a structure called as bulbils. These bulbils, they arise on the lateral side of the stem, and upon arriving the comfortable and convenient circumstances, they germinate and give rise to a proper leaf and other systems. Now, in general, gymnosperms are heterosporous. That means they produce two kinds of spores, and these two kinds of spores are produced in two different spore bearing structures. The spore bearing structure, which contains the smaller size spores, they are called as microsporangium or the pollen sac, whereas the larger spores are contained within megasporangium, also termed as ovules. In case of gymnosperms, here is a typical representation of a microsporangium and a megasporangium, as you can see in the diagram. A microsporangium is represented by a single intact layer of tissues called as integuments, which contains some of the diploid cells called as nucellus, and inside the nucellus is found one megasporocyte. Whereas the microsporangium is found on the lower side of microsporophylls. The structures which bear these microsporangia on their lower side, they are termed as microsporophylls, and the cluster of these such microsporophylls, they are called as microstrobilus. On the contrary, these megasporangium, also called as ovule, they are found on the adaxial side of megasporophyll, and the cluster of these megasporophyll, they are termed as megastrobilus. The gymnosperms can be monoecious, such as Pinus or Cedrus, or they can be dioecious also, such as Cycas, Ephedra, Jingo, etc. In dioecious species, the male and female cones are borne on separate plants. Here you can see in the diagram that Pinus tree bears female and male cones on the same branch. So here this is an example of a monoecious genus, whereas in case of Cedrus, the male and the female cones, they are borne on different plants. In case of Cycas also, which is a look-like of a fern, but they are not either a fern nor they are a palm tree, you will find that the cones are borne on different plants. So the plant which bears the female cone, they are called as female plant, whereas the the plant which bears the male cone, they are called as the male plants. The position of the strobili on the plant varies from terminal to lateral. Microsporangia are born on the abaxial or the lower surface of the microsporophyll, whereas ovules, that is the megasporangium, they are generally born on the adaxial or the upper surface of a leaf-like structure called as megasporophyll or ovuliferous scales. Now, for the sake of some clarity, let us understand the basic concept of adaxial and abaxial side with the help of a small diagram. So, in the diagram you can see that there is a central stem axis, and on the lateral side of this central stem axis arise a leaf. So, a leaf will have 4 different ends. The outermost end is termed as a distal end, which is towards the axis is termed as proximal end, and the upper side of the leaf pinna, that is called as adaxial side, and the lower side is termed as abaxial side. In case of gymnosperm, the spore mother cells contained in the sporangia, they undergo meiosis to form microspores and megaspores respectively, and they represent the first cell of the haploid and gametophytic generation. The ovule is generally orthotropous. During the development of the microsporangium, the initials form archisporal tissues, form wall layers and sporogenous cells. These sporogenous cells, they later give rise to microspore mother cells. The microspore mother cell or MMCs, they divide meiotically to form tetrads of microspores. This development can be seen with the help of a diagram. Here, from a male cone, this is a scale of pollen strobilus. Within this, on the lower side lies microsporangia, and within this microsporangia, microspore mother cells are found. Each microspore mother cells, they undergo meiosis to form 4 microspores. So, these 4 microspores are the first cell or the first representative of gametophytic generation in gymnosperms. Individual microspores, they metamorphose into specific structure now termed as pollen grains. Each microspore undergoes mitotic division to form 1 or more prothallel cells and a single entheridial initial. This entheridial initial finally forms the male gametes. The development of male gametophyte is endosporic in gymnosperms. That means it takes place partially in the microsporangium and partially in the pollen chamber of the ovule. At the time of shedding, the pollen grains are usually multicelled and contain 1 or 2 or more prothallel cells and a tube nucleus and entheridial cell. So, overall, the male gametophyte is a multinucleate or multicellular structure. The male gametophyte in gymnosperms can be represented with the help of these diagrams. Here, typical pollen grain of Pinus is shown. Here we can see that there are 4 microspores. Each microspores, they have few prothallel cells, 1 entheridial initial and then the wing cells. The prothallel initials gradually degenerate and the generative cell is the one which gives rise to male gamete proper, and the anterior-most cell gives rise to the pollen tube, or this is also termed as tube cells. So, this entire structure which contains a pollen tube, spermatogenous cells and sterile cells, they are collectively termed as male gametophyte. In case of Cupressaceae, Taxodiaceae, Cephalotaxae and Taxaceae, the pollens are shed at uninucleate stage and the gametophyte development continues after pollination. In case of Pinus, the pollen grains are winged and similar conditions are found in Podocarpus, whereas in case of Cycas and Jingo, the pollen grains are non-winged. The male gametophyte either lacks a prothelial cell, as the case is found in Taxodium and Cephalotaxus, or its number ranges from 1 as it is found in Cycas to 32 as it is found or it can be seen in Agathis and Araucaria. The male gametes are flagellate in case of Cycas and Jingo, and they are non-flagellate in case of Pinus and Cedrus. Now let us talk about the female gametophyte, which is contained within an integumented structure called as ovule or the megasporangium. As we all know that ovules are naked and they are borne on the adaxial surface of the megasporophyle. When we talk about naked ovules, that means they are not enclosed by megasporophyle or the subtending structures. Only one single integument covers the entire structure. These ovules are generally arranged spirally around a central cone axis. This central cone axis sometimes turns woody in some of the coniferous structures, and these central cones, these megasporophyll bearing structures, they are also termed as megastrobilus or female cones. The ovules are sessile. They happen to be sessile and orthotropous, and each of these ovule consists of a parenchymatous mass of cells. These parenchymatous mass of cells are termed as nucellus or megasporangium. The nucellus or the megasporangium, it becomes enveloped by a single massive integument that grows around it, leaving a small opening or the micropyle at the tip. Now, when we talk about the orthotropous ovule are such ovules which have the micropyle and chalaza on the same axis, and the micropyle and the chalaza, they are found on opposite poles or opposite ends. So, the ovule structure can be seen with the help of this diagram, where a female cone in LS looks like multiple leaf-like structure, and all of these leaf-like structure on its adaxial side contains two ovules generally, and these ovules, they get fertilized to produce seeds, and these seeds, they have long appendages also termed as wings. The megasporangium enclosed by the integument is called the ovule. That means the nucellus, if enclosed with the help of a thick integument, this is nothing but your ovule. The nucellus contains a single diploid megaspore cell. These single megaspore cell which are diploid, they undergo meiosis and they form the tetrad of haploid megaspores, of which only one remain functional while the other three degenerate. Now, this can be seen with the help of, this can be understood with the help of this diagram, where you can see that a parenchymatous mass of cells, also called as nucellus or megasporangium, is covered by a single layer of integument. Now, inside this megasporangium lies one single megaspore. This single megaspore, it undergoes meiosis to give rise to 4 haploid cells. Out of these 4 haploid cells, 3 degenerate, and here we can see only one megaspore. This megaspore is always surrounded by spore wall, and within this spore wall, this megaspore undergoes multiple mitotic divisions, and these mitotic divisions make this entire structure a multinucleate female gametophyte. So megaspore represents the first cellular generation of female gametophyte. The functional chalazial megaspore enlarges and undergoes free nuclear division, and thus the young gametophyte develops within the megaspore, and which is enclosed by spore wall. It is very much clear in this diagram where we can see that a megaspore which is covered by spore wall undergoes multiple divisions and give rise to the female multinucleate multicellular female gametophyte. This female gametophyte is surrounded by nucellus. There is a division in centripetal formation which starts around the free nuclei of the female gametophyte, which goes on until the whole gametophyte becomes cellular. Now this is the female gametophyte which also serves as endosperm. So the endosperm in case of gymnosperm is always haploid in nature. So this is also one of the very stark difference between the development of endosperm from angiosperm and gymnosperm. In case of gymnosperm, the endosperm happens to be haploid in nature, whereas in case of angiosperm, as we all know, the endosperms are produced after double fertilization, and the level of ploidy is triploid in each of the endosperm cells. Now let us talk about the apical region of the ovule. The apical region of the nucellus degenerates to form a pollen chamber in which pollen grains will come and lie until their further growth. The female sex organs or archegonia, they are developed towards the micropylar end of the female gametophyte. In case of Netum and Welwitschia, they lack a very distinct archegonia, but yes, the cells are almost similar to archegonia only. This can be seen with the help of this diagram, where the nucellar cells, they degenerate at the micropylar end to form a pollen chamber. Now this pollen chamber will host pollen grains. Just below the pollen chamber, the archegonia are found within the female gametophyte. So there is a female gametophyte surrounded by nucellar cells, and here is the pollen chamber. These archegonia in gymnosperm are much reduced. They are much reduced, and sometimes they are almost ephemeral. The only function of these archegonia is to provide protection to that one single egg cell which will get fertilized. This is an enlarged picture where you can see very clearly that through the micropyle the pollen grains will come to the pollen chamber, and two archegonia will host these antherizoids and they will get fertilized. Now let us talk about the pollination process and fertilization. Pollen grains are carried to the ovule, and this is followed by fertilization. The male gametes are carried to the egg by means of the pollen tube. Syngamy, or as we also call them fertilization, between two haploid gametes, that is male gamete and female gamete, restores the ploidy level of 2n. The new sporophyte is first represented by the zygote that later on develops into embryo. Thus, the cycle is completed. There is distinct alternation of generation in case of gymnosperms also. The sporophytic generation predominates the entire life cycle, whereas the gametophytic generation is represented by few cell structure of male gametophyte and female gametophyte, also enclosed within ovule and the male gametophyte enclosed within the microsporangium. The pollination in gymnosperms is siphonogamous. Siphonogamy is a term used to collectively designate plants in which the sperms are directly transported to the egg by means of a pollen tube, and it usually takes place by means of wind, that is anemophily, and abundant pollen production ensures such type of pollination. In case of cycas, ephedra, netum and welwitschia, entomophily has also been reported, where the insects play the part of pollinators. During the process of fertilization, a pollination drop is secreted by the ovule at the tip of the micropyle, and this captures the wind-borne pollen grains. The partially developed male gametophytes, they are drawn or sucked into the ovule by the drying of these pollination drops. These pollination drop exudates not only receives the pollen but also places them very close to the nucellus. The pollen now lies in the pollen chamber and on the nucellus in the close proximity of the archegonia. The entire life cycle can be represented with the help of this diagram, where you can see that from the female cone and male cone, male pollen grains and female structures, they come together, they fuse, meiosis takes place, the megaspores are formed, the eggs are formed, and pollen tube is formed. This pollen tube fertilizes and zygote is formed, and then mature seed is formed. And then from these mature seeds, after germination, a new sporophytic generation is born. In the gymnosperms, pollination may not be immediately followed by fertilization. Sometimes what happens, a very long interval elapses between the two phenomena. During this intervening phase, also called as the resting phase, the ovule shows many changes and gets ready for fertilization. The micropyle is closed and the ovule enlarges in size. The pollen grains on reaching the nucellus germinate immediately or after a resting period. The pollen tube reaches the female gametophyte and finally makes contact with the neck of the archegonium. The formation of seed is the result of fertilization and consequent enlargement of the ovule. The zygote develops into an embryo and the endosperm is formed from the persisting female gametophyte, which provides nutrition also. The nucellus gets disorganized and is visible at the micropylar tip of the seed as a thin cap. The integument forms the seed coat. The seeds, they are a remarkable combination of two sporophytic and one gametophytic generation. The seed coat is the old sporophyte, the embryo is the new sporophyte, and the endosperm is the persistent female gametophyte. The number of cotyledons varies from 2 to multiple. The detached seeds of gymnosperms remain dormant and undergo a prolonged period of rest. With the onset of favorable conditions, the embryo again resumes its growth by rupturing the hard seed coat and develops into a new plant. The germination in case of gymnosperm is always epigeal in most of the genera. That is, the cotyledons, they emerge above the ground. In case of cycas and jingo, the seeds show no dormancy. They germinate immediately on falling on the suitable substrate, or else they lose their viability. You can see in the diagram that how in case of epigeal germination, the cotyledons first come out of the substrate level and then the seeds give rise to various leaf and other structures of the plant. So this was all about the reproductive aspects in case of gymnosperm. We have seen the male and female reproductive structures, sex organs, how the fertilization and pollination takes place, and then finally the germination takes place and a new sporophytic generation is produced. I hope I have been able to cover up the reproductive aspects of gymnosperm in much detail. In our next lecture, we will see the economic aspects of gymnosperm in a much broader detail. Thank you so much.